Okay, in today's lecture we will discuss about cell banking. A cell bank is basically a facility that stores cells of specific genome for different purposes. The first person who is credited with this, with making cell banks for widespread use, was Krell, who was a scientist who created his first cell bank collection in the late 1890s. Bank is basically the collection of appropriate containers whose contents are of uniform composition. These are stored in under defined condition, and each container represents an aliquot of single pool of cells. These could be master cell banks, or called as the MCB, or working cell banks. Master cell banks are basically the collection of cells of uniform composition which are derived from a single source, while working cell banks are derived from one or more vials from the master cell bank and used for the production. There are different sources for the cell banks. These sources could be of human origin, could be of animal origin, or could be of insect origin. And there are different categories of cell banks. First is the primary cell culture, diploid cell culture, continuous cell lines, and stem cell lines. So here is the procedure in which you can make the cell culture. First you will derive, or you will isolate the cells from the source, and then you can make primary cell cultures, or from that primary cell cultures, with the passage of time, you can make diploid cell lines, or after passage of time you can also make the continuous cell lines, or which we also called as the immortal cells. Primary cell cultures, majority viral contaminations are made are used or are stored as primary cell cultures. Advantages of making these primary cell cultures are that they are comparatively easy to prepare using simple media and bovine serums. They generally possess broad sensitivity to a variety of viruses. Disadvantages include that the contaminations of infectious agents is higher risk than with DCLs and CCLs. The quality and viral sensitivity of cultures which are obtained from different animals are variables, so primary cell cultures cannot be tested as extensively as DCLs or CCLs. DCLs, for example, if they are from human or monkey origins, a finite capacity for serial propagations, and they remain alive and metabolic active, but may show some morphological or biochemical changes with the passage of time. So, though they are non-tumorigenic, they display diploid cytogenetic characteristic with a low frequency of chromosomal abnormalities. Advantages which DCLs includes, they are well characterized and they are standardized, cryopreserved cell bank systems that allow for consistency and reproducibility, and these are non-tumorigenic. While disadvantages which are associated with DCLs are they are not easy to use in large scale productions, more fastidious, nutritional requirements are more than the other cell substrates, and they are difficult to adapt in the serum free growth. When we talk about CCLs, these are the serial subcultivations of primary cell cultures of animal tumors or animal cells. These are transformed from the normal cells with oncogenic virals or viral sequences. Serial subcultivation of primary cell productions derived from normal tissues that having an apparently indefinite lifespan. Advantages which are associated with CCLs are they are well characterized and standardized, and they can be cryopreserved, and that allows for consistency and reproducibility, grow more easily than DCLs, and can easily adapt to serum free growth, and can grow on microcarriers or small scale productions. Disadvantages that associated with CCLs may express endogenous viruses, and some are tumorigenic. in immunosuppressed animal model, and theoretical risks identified by nucleic acid transforming proteins and viruses. Potential risks and risk mitigations. Virus and other transmissible agents are the potential risk. Cellular nucleic acids and growth promoting proteins are basically the potential risks and risks which are associated with the cell banking.